Okay, good evening everyone. So we'll start with today's class. Other people can keep joining. So today we are going to discuss a set of 12 questions for the lecture material provided for week 11. So we have already discussed questions for 10 weeks and this is the 11th week. We have 1 week left. It is the 12th week and then this live session is completed for the subject Industrial Biotechnology. Okay? So, if you all have any kind of doubts, you can ask me in the live session or you can comment it the YouTube videos and we can discuss your doubts in the class itself. Okay? Because the live sessions are coming to an end. If you have any doubts and queries from the live sessions, first priority is the live sessions. So if you have any doubts from the live sessions, then you are most welcome to ask. And secondly, as I'm not sure how much good response you are getting on your doubts for the lecture material or current year assignment questions, I can help with that also if you really want to solve any kind of problem from the current year assignments. Okay, that also we can discuss. But yeah, right. If you have any doubts, you should ask today so that if I am so that I can be prepared and answer your queries in the next session, right? So if you have any kind of doubts, please feel free to ask. And it's already 7:10, so we'll start with today's session. We have 12 questions, and I hope everyone knows the rules. So you have be shown the question, then you will be given around 3 minutes time, and then you can type your answer of choice in the chat box and then we'll discuss the correct answer to it in some time, the explanation for it. Okay, so let's start and please keep yourselves mute throughout the session. Okay, if you have any doubts, write it down in the chat box. So let's start with first question of today. So this for this week the questions are a bit lengthy, so try to read the questions and options properly and then answer it. Okay, which of the following is not considered one of the principal categories of biopesticides described in the lecture? Microbial pesticides, plant incorporated protectants, botanical pesticides, organochlorine, synthetic insecticides. Start answering. Okay, let's see what is the correct answer to this question. Done the answers. So it's D, organochlorine synthetic insecticides. Okay. So it was asked which is not considered one of the principal categories of biopesticides described in the lecture. So it is the organochlorine synthetic insecticides. So according to the lecture material given, biopesticides are classified into microbial pesticides, PIPs, that is the plant incorporated protectants, biochemical pesticides, botanical pesticides, and biotic agents, that is predators or parasitoids. So organochlorine synthetic insecticides are conventional chemical pesticides. These are not biopesticides. So they do not come in the biopesticide category. So okay, Shanmupriya has given the correct answer. Moving on to the next question. What is the primary lethal mechanism by which Bacillus thuringiensis formulations kill susceptible insect larvae? A. Disruption of the insect nervous system by blocking sodium channels. B. Release of crystal toxins that damage the midgut epithelium after ingestion. C. Inhibition of chitin synthesis causing failed molting. D. Sterilization of adults by endocrine disruption. Start answering. Okay, let's see what is the correct answer to this question. Uh, is B. Release of crystal toxins that damage the midgut epithelium after ingestion. So, what was the question? What is the primary lethal mechanism by which the BT formulations kill susceptible insect larvae? So, what they do, they release these crystal toxins that damage the midgut epithelium after ingestion. So, they damage the epithelium of the insect larva by releasing the crystal toxins which is taken up by the insect larva. So explanation is written as BT spores or crystal proteins are toxic when ingested by the larva. The crystal tox proteins bind midgut receptors, form pores and damage the midgut epithelial cells leading to larval death. Okay, this was quite clear. On to the next question. Agrobacterium radiobacter strain used commercially suppress crown gall disease primarily by reducing a bacteriophage that kills A. tumefaciens, uh, producing an antibiotic agrocin that is toxic to A. tumefaciens and competitively colonizing the roots. C. Inducing systemic acquired resistance in host plants through salicylic acid production. D. Secreting cellulase enzymes that break down A. tumefaciens cell walls. Start answering. Read the question and the options properly and then only answer it. Okay. Okay, let's see what is the answer to this question. It is B. Producing an antibiotic agrocin that is toxic to Agrobacterium tumefaciens and competitively colonizing the roots. So, how do these bacteria These Agrobacterium radiobacter suppress the crown gall disease. They produce an antibiotic which is called as the agrocin, which is toxic and competitively colonizes the roots. So the explanation is written as the effective strains of the Agrobacterium radiobacter colonize roots and produce agrocin, an antibiotic active against Agrobacterium tumefaciens, thus preventing the crown gall. Both colonization and agrocin production are important features. Okay, on to the next question. Which entomopathogenic microbes correctly matched to the pests it commonly controls as given in the lectures? So A is Metarhizium anisophyllae, spittlebugs and rhinoceros beetles, B Trichoderma, aphids and whiteflies, C Pseudomonas fluorescens, Colorado potato beetle, D Optreillae, damping of pathogens, Pythium species. Start answering. Okay, let's see what is the correct answer to this question. It is A. Metarhizium anisopleum. So that is A. Citralbugs and rhinoceros beetles. So the correct match here is the A option. Okay. The other pairings are incorrect because trichoderma is used against soil-borne fungal diseases. Deflorescence is used for controlling damping of pathogens, and Nomure relay controls certain caterpillars. So other matches are wrong. First one is correct. On to the next. Which statement about baculoviruses BVs is inconsistent with the lecture slides? So these questions are made keeping in mind what material is given in the lecture slides, so you should be thorough with it. Okay, they are broad spectrum bacterial pathogens used against nematodes. Their genome is single stranded RNA and they are enveloped spherical viruses. They control lepidopteran and some hymenopteran pests and contain circular double stranded DNA. Virions are rod shaped. They function via systemic toxin production in plants. Start answering. Yeah, yeah, you can leave some urgent. Okay? But this is like shit. So it's only Shanpriya now. Okay, follow me forward with you, Shanpriya. Okay, let's see what is the correct answer to this question. It is C. They control lepidopteran and some hymenopteran pests and contain circular double-stranded DNA. Virions are rod-shaped. So the explanation given is the baculoviruses target the Lepidopterus and Hemipterus pests and describe them as rod-shaped and circular double-stranded DNA genomes. These are the features used in biological control of insect pests. So this statement is correct about the baculoviruses. Okay, it's quite clear. Moving on to the next. Which of the following statement about anaerobic digestion is correct? A. Anaerobic digestion converts organic matter primarily into oxygen and organic acids. B. The process produces methane and carbon dioxide, and the digested slurry can act as a soil amendment with measurable NPK. NPK values are given. C. Anaerobic digestion destroys all macronutrients, so the effluent is nutrient free. D. The process only produces methane, no carbon dioxide is formed. Which of the following statements are correct about anaerobic digestion? That is the question. Okay, read the question properly. Answer it. Okay, let's see what is the correct answer to this question. It is B. The process produces methane and carbon dioxide, and the digested slurry can act as a soil amendment with measurable NPK. Okay, moving on to the next question. Which microorganism is commonly reported in the lecture as an industrially used PHA producer? Cupria vitis nectar, formerly, okay, no need to read that name. B. Saccharomyces cerevisiae. C. Staphylococcus aureus. D. Influenza A virus. So, which microorganism is commonly used as a PHA producer? Okay, that is the question. Start answering. Okay, let's see what is the correct answer to this question. Is A. Cupria vitis nectar. So the explanation is among the organisms used for economical, these are the organisms which are mainly used for economical PHA production, whereas yeast like Saccharomyces cerevisiae and others like Staphylococcus and influenza A viruses are not typical industrial PHA producers. So the correct option is A. Moving on to the next question, which of the following substrates is least typically cited in the lecture as an economical feedstock for PHA production? Sugarcane molasses, excuse me, glycerol, a byproduct of biodiesel, cheese waste stream, virgin petrochemical ethylene, pure petrochemical feedstock. Start answering. Okay, let's see what is the correct answer to this question. It is D, virgin petrochemical ethylene, which is the pure petrochemical feedstock. So this is the one which is least used as an economical feedstock for FH PHA production because it is a petrochemical feedstock. So there are many agro-residual and renewable substrates listed as economical PHA feedstocks in the lectures, like molasses, glycerol, cheese whey, starch, hydrolysates, or oils. Whereas the pure petrochemical raw materials are not emphasized as economical or renewable substrates, right? Moving on to the next question. For typical laboratory or industrial PHA production, which fermentation conditions are most frequently used? A 5 to 10 degrees and pH 3 to 4, B 30 to 35 degrees and pH 6.5 to 7, C 60 to 70 degrees and pH 9 to 10, D 100 degrees and pH 1. Start answering. Okay, let's see what is the correct answer. It is B, 30 to 35 degrees and 6.5 to 7. This is the optimal fermentation conditions for industrial PHA production. On to the next question. Which pair of glass transition temperature and melting point best matches polyacetic acid PLA? So first is the transition temperature is 50 degrees and melting temperature is 0 to 10 degrees. Next is transition temperature is 50 to 60 degrees and melting temperature is 130 to 180 degrees. C is transition temperature 200 to 250 degrees and melting temperature 300 to 350 degrees. D, transition temperature is 0 to 5 degrees and melting temperature is 20 to 30 degrees. Start answering. let's see what is the correct answer to this question. It is B. Tg is equal to 50 to 60 degrees and Tm is equal to 130 to 180 degree. So these are the temperatures which best matches with the polylactic acid. Okay, so these are the thermal characteristics which explain that the PLA's applications and its brittle nature, that is low elongation. Moving on to the next question, according to the lecture, which expression system and downstream assembly step are characteristic of current commercial hepatitis B vaccines? A. HBsAgs, that is the hepatitis B vaccines produced in E. coli assembled into capsids by viral proteases. B. Vaccines produced in yeast assembled into nearly 22 nanometer lipoprotein particles and absorbed to aluminum hydroxide as adjuvant. C. Whole inactivated HPV grown in eggs and formulated with mercury only. D. HPV's AG chemically synthesized as a 200 kilodalton polymer and delivered without adjuvant. Start answering. For the essay type questions, I think you should be thorough with the theory. Uh, I have no idea which kind of questions they may ask because I am not in charge of those exam questions. I can only say that you should be thorough with each and every topic discussed in the lectures, or at least the topics from which the current year assignment questions are coming. I think those are the important topics from which you should practice as well. Okay. Okay, let's see what is the correct answer to this question. Is B. HPSAG, that is the hepatitis B vaccines in yeast Saccharomyces cerevisiae assembled into nearly 22 nanomolar lipoprotein particles and absorbed to aluminum hydroxide as adjuvant. These are the ones. This expression system and the downstream assembly steps are characteristic for the hepatitis B vaccines. Okay. The last question of today's session: which reagent or process is used during early insulin manufacturing, recombinant production in E. coli to separate the insulin genes before forming the correct disulfide bonds? A. Cyanogen bromide cleavage and methionine residues followed by oxidation to form disulfide bond. bonds. B. Trypsin digestion followed by glycosylation and yeast. C. Heat denaturation at 150 degrees Celsius to break peptide bonds. And D. RNase E treatment to remove contaminating RNA. Okay, start answering. Okay, let's see what is the correct answer to this question. It is A. Cyanogen bromide cleavage at methionine residues followed by oxidation to form disulfide bonds. So, the A and B chains in the E. coli and using cyanobromide, which cleaves the methionine residues, release the insulin chains, and then these chains get combined and oxidized to reform the native disulfide bridges. So that's why cyanobromide Cyanogen bromide is used to cleave the methionine residues that is followed by oxidation to form the disulfide bonds. Okay, so that's it for today's session. Shanmukhpriya, can you tell me your score out of 12? Okay, that is a good score. Uh, I will suggest you uh, to use AI, anything, either ChatGPT or Perplexity, whatever you can use. Attach your week's file in it and tell the AI to make for you the revised notes in a very concise manner. Okay, I think that is the only way you can shorten it and revise it once more. Other than that, you have to make particular notes, but you have said that you have not made it. So let AI do the work and tell it to make brief notes for each week, attach each week's lecture material, and tell him to make short notes on all the important topics of the lecture. And do that. Or if you want, you can attach your, that will be better. That's the thing I can suggest you. Revise all PDFs. When we have technology, I think we should make use of it. But what do you think? Yeah. Okay. Thank you, Shanmukta, for joining and being throughout the session. So next week, Tuesday, is the last class. Make sure you join, and if others join, that is well and good. You should join. Okay. Keep up the keep up with the consistency, and I wish you well that you score good in the exams. Okay. Okay. Thank you. Bye.